Hi, welcome to my presentation of Chapter 10 on muscle tissue and muscle physiology. So you have three types of muscle tissue: skeletal muscle, cardiac muscle, and smooth muscle. Each of them has similarities and differences. For each of those types of tissue, a muscle cell is also called a muscle fiber or a myocyte. The prefix myo means muscle, and then the ending cyte just means cell. So a myocyte is just a muscle cell. A muscle fiber is also a muscle cell. For your skeletal muscle, that's the main type of muscle that you think about. That is the muscle that allows you to move. Most of your skeletal muscles are connected to bones by tendons, but you also have some that are attached to other things like skin, mostly attached to bones though. The skeletal muscle is under your voluntary control, so you choose when to move it. Structurally, it is striated, which means that if you look at it under the microscope, you'll see little lines going across each cell. Those are the striations. Those are related to the microscopic structure of the cell. This is related to the contractile organelle within the cell. Muscle cells, skeletal muscle cells also have multiple nuclei. So each of these blue or kind of purplish things is a nucleus. They're really long cells that have many nuclei, not just one. For your cardiac muscle, that's only found in the heart. It is also striated, so it also has the little lines going across. But the cells are much shorter than the skeletal muscle cells, and they branch. And they're all connected to each other at intercalated discs. So this would be one cardiac muscle cell, and you can see it's branching, and then it's connected to other cardiac muscle cells at these intercalated discs. At those discs, you have gap junctions that actually connect the cytoplasm of the different cells together, and that lets them all contract together as one unit, which you need to have a powerful heartbeat. Cardiac muscle is involuntary, so you cannot consciously control it. Also, it's the only type of muscle that is able to contract without stimulation from the nervous system. So it's kind of self-stimulating, and even if you were brain dead, your heart would continue to beat. The nervous system is just able to speed the heart rate up or down as necessary. And then the last type of muscle is a smooth muscle. You find that in a lot of different organs. It'll be found in the walls of organs very commonly, like the stomach and the intestine and the blood vessels, the bladder, all over the place really. The smooth muscle does not have striations. That's why it's called smooth muscle. So as you see, you don't have any of the little lines going across it there. A lot of the time, smooth muscle cells will have gap junctions connecting them, but not always. It just depends on where it is and what it's doing. If you do have gap junctions, then that makes the smooth muscle able to contract as a single unit. And smooth muscle is always involuntary, so you're never able to consciously control it, even though the nervous system does need to stimulate it to contract. It doesn't contract by itself. So for most of this presentation, we're going to be talking about skeletal muscle, and then at the end, we'll talk about some of the differences between skeletal and cardiac and smooth muscle in more detail. For all types of muscle, skeletal, cardiac, and smooth, all of those tissue types share these 5 characteristics. First, contractility. So all muscle cells are able to contract. That usually means that the whole cell will get shorter, and is based on proteins inside of the cell that are able to move past each other and towards each other, sort of interlocking like if you were to interweave your fingers. All muscles are excitable, which means that the muscle cells can respond to stimuli. Usually that means that they respond to neurotransmitters, but some types of muscle can also respond to other things. And that response is a change in their membrane potential. So they're similar to neurons in that respect, where they have a membrane potential that will change once they've been stimulated. They also have conductivity. which means that they can basically carry action potentials. If you have a change in the membrane potential of a muscle cell, that can spread across the entire cell membrane as an action potential. So that's also like neurons. And then you have distensibility, which is the ability of muscle cells to stretch longer than they normally are. And then finally, elasticity, which is the ability to return to their original shape after they've been stretched. Usually we think of elasticity as meaning that something is stretchy, but technically it just means that if it has been stretched, it can return to its original shape. Stretching itself is distensibility. So again, even though all muscle cells share these five characteristics, most of this presentation will only be talking about the skeletal muscle tissue. And I probably won't always say skeletal muscle, but it always is skeletal muscle until the very end. So, skeletal muscle cell structure. Within a skeletal muscle cell, you have most of the organelles that you would ordinarily have, but you also have some extra organelles, and some of the regular ones have a different shape. So the main organelle that you don't have in other types of cells is the myofibril. The myofibrils are long kind of columns of proteins, of contractile proteins. So this is a myofibril, this is a myofibril, this is a myofibril. The myofibrils would be stretching the entire length of the skeletal muscle cell, and they contain the proteins that are going to be moving towards each other to contract the cell and allow it to shorten its length. So most of the volume of a skeletal muscle cell is actually occupied by these myofibrils. Then you have kind of special names for some of the cell parts of a muscle cell. So for the cytoplasm, which is just all of the stuff inside of the cell except for the nucleus, in a normal cell that's called the cytoplasm, but in a muscle cell we call that the sarcoplasm or the myoplasm. Myo means muscle, of course, and then sarc means flesh, and we generally associate muscle with, well, the meaty part of the meat is flesh. The cell membrane of a muscle cell is called the sarcolemma or the myolemma, so that also has a special name. The nuclei of a muscle cell are not in the center of the cell where they would be in a normal cell. Instead, they're pushed off to the side there. They've been kind of crowded out by all the myofibrils. So here you have a normal cell compared to this muscle cell, skeletal muscle cell. So of course a skeletal muscle cell is much bigger and longer. The normal cell has the nucleus in the middle, not on the side. The normal cell does not have any myofibrils in it. It has a cytoplasm that includes everything except the nucleus, and it has a plasma membrane around it, whereas the muscle cell has a sarcoplasm, which would be everything except for the nuclei, and it has a sarcolemma around the outside of it. Then you have the sarcoplasmic reticulum, which is a specialized type of smooth endoplasmic reticulum that you would have in a normal cell. So in a normal cell, the smooth endoplasmic reticulum is involved with synthesizing lipids and transporting them to where they need to go. In the muscle cell, it has a different role. It stores calcium. In a regular cell, the sarcoplasmic reticulum would be found around the nucleus of the cell, but in a skeletal muscle cell, you find it wrapped around the myofibrils. So that would be all this blue stuff here. All of that is the sarcoplasmic reticulum, and it is storing calcium ions. You also have little tubes or tunnels, little tunnels that run down from the sarcolemma into the muscle cell. Here you can see the opening of one, here's the opening of another. So those would be attaching to the actual sarcolemma. Here you have openings of some on the actual sarcolemma. And they kind of go down into the cell and then start running around the myofibrils with sarcoplasmic reticulum on either side of them. And those T tubules end up being really important for muscle contraction, so we'll come back to them later. They are kind of open to the outside, so they would be filled with the extracellular fluid. If you consider the T-tubule with the sarcoplasmic reticulum on either side of it, that is considered a triad. So your T-tubule in yellow here, along with this thickened sarcoplasmic reticulum on either side of it, that's a triad. If you were to talk just about the thickened part of the sarcoplasmic reticulum that is on the side of each T-tubule, that is called the terminal cisternae. So just to look at it again, here you have the myofibrils that are organelles unique to muscle cells that allow contraction. Here you have in blue the sarcoplasmic reticulum, which would be the smooth endoplasmic reticulum in a normal cell. Its job is to store calcium ions that the cell needs to contract. Then you have these T-tubules running along the myofibrils, and they also open to the outside of the cell. The T-tubule with the sarcoplasmic reticulum on either side of it is a triad. There's a triad. And this thick part of the sarcoplasmic reticulum that's on either side of the T-tubule is called the terminal cisternae. Then you have the nucleus on the side of the cell. You also have some mitochondria in there. And you're going to have multiple nuclei in these cells. So there's one and there's one. And they're really long cells. Okay, so within each myofibril, you have myofilaments that make it up. Each myofilament is like a chain made of different proteins. You have three types of myofilaments: thick, thin, and elastic. The thick filament is right here. The main protein it's made of is myosin, which is this little guy right here. You can see it has this head on the end, so a bunch of those together makes the thick filament with all the little heads sticking out at each end. This is a thin filament. The main protein it's made out of is actin. The thick and the thin filaments together are providing the force for contraction. And then the last filament is the elastic filament. It's made out of the protein titin. As the name suggests, it can stretch and then bounce back to its original shape. And its role is structural. So it's linking the thick and thin filaments together and also just providing strength so that the myofibril does not fall apart. If you look at the myofilaments are arranged in regular patterns, all running parallel to each other. You'll have a band with thick filaments in it. Then on either end, you'll have thin filaments that are partly overlapping with the thick filaments. And then you also have these elastic filaments that are connecting the thick filaments to the thin filaments that are kind of up and down from them. And then you also have the other thick filaments that are on the side of the thick filaments. So in muscle contraction, these thin filaments would actually be sliding in towards the center of the thick filaments, so that this whole thing would be getting shorter. The whole myofibril would be getting shorter. If you have one of these bands of the thick filaments and then the two bands of thin filaments on either end of it, that whole thing together is a sarcomere. A sarcomere is the functional unit of the myofibril that allows for muscle contraction. So every sarcomere contains everything you need to have muscle contraction. The thick filament, the thin filament, they can slide past each other, and then you have the elastic filaments holding everything together. Also, this arrangement of filaments in different bands is what creates the striations that you can see under the microscope if you look at a skeletal muscle cell or a cardiac muscle cell. So we'll go into more detail on each of these myofilaments. First, we'll talk about the different proteins in them. You have 5 different proteins that compose these myofilaments. 2 of them generate force. Those are myosin and actin. Then you also have two more that are regulatory proteins. Those are tropomyosin and troponin. So the myosin and actin are acting together to create force that allows the muscle to contract. And then the tropomyosin and troponin are controlling how that happens or when that happens. And then finally you have titin, which is a structural protein. It's basically just making sure that the myofibril stays together and doesn't fall apart. So for each of these filaments, we'll go through its structure, starting with the thick filament. It's mostly made out of myosin. This is one myosin protein. So it has kind of a long tail and then a fat head at the top. The head is actually able to bend. So right where the head joins to the tail, you have a hinge and the head can bend there. So the thick filament is mostly made of those myosins, so that the heads are sticking out at either end of the filament. In the very center of the filament, you'll also have titin, giving it structural support. Then next you have the thin filaments. Those are mostly composed of actin. Each of these little purple things is an actin, and each actin has an active site where the myosin head can bind it. So for the muscle to contract, you would have a myosin head grab this actin active site, and then the myosin head would bend at the hinge, which pulls the two filaments together. Normally, the actin active sites are going to be blocked by the other proteins in the thin filament, the tropomyosin and the troponin. So the tropomyosin is this long blue protein here, and you can see it's arranged that it's on top of the actin active sites. And then you have troponin, which is anchoring it in place. So in order for the muscle to actually contract, you have to move this troponin and the tropomyosin out of the way so that the actin active sites are exposed, and then myosin can bind them. Then finally, your elastic filaments are nothing but titin, which as you can see is coiled up, which makes it elastic so that when it stretches, it will bounce back. And that just gives your muscle structural support, or the myofibril structural support. So you can see that these elastic filaments are linking the thin filaments to the thick filaments that are past them, which just keeps the whole thing together. We're actually going to skip this. So you have all these different bands that are visible if you have a fancy microscope on the muscle. Those are created by the pattern of myofilaments within each myofibril, but we're not gonna go into the details on those bands. So we're gonna skip this too. Okay. So I guess we actually didn't go over some of this. It's from chapter 9, which we skipped, so I'm just gonna skip this as well. No, I'll just go over it real fast, because some of it actually is from chapter 10. So there's a lot of M words in the muscles, a lot of myo this and myo that. It can get confusing. So the whole thing, the whole muscle, if you were to look inside of it, you would find fascicles, which are bundles of the muscle cells, and then within that you would find individual muscle cells, which are also called myocytes or muscle fibers, and that's where we've picked up in chapter 10. The fascicles would be talked about in chapter 9. But these individual muscle cells, the myocytes, if you zoom in on them, you have the myofibrils within them, which are the contractile organs. If you zoom in on the myofibril, then you get to the myofilament proteins that compose it. So you have myofilaments at the lowest level, then those compose myofibrils, which are contractile organelles, and then those compose myocytes, which are the actual muscle cells that are also called muscle fibers. Okay, so now we'll go through how contraction actually occurs. So the dominant theory of skeletal muscle contraction is the sliding-filament theory, which is basically saying that when the muscle contracts, your thick and thin filaments move towards each other. So actually the thin ones would be moving in towards the thick ones. until they get close to the center, and then they can't move anymore. And it's basically like your fingers. If you were to interweave your fingers and then move them closer together, you can imagine that on one hand you have the thick filaments, on the other hand you have the thin filaments, and then they're moving together. The way that they move together is that the myosin heads on either end of the thick myofilaments would be grabbing onto actin-active sites and then turning, bending at the hinge, which ends up pulling the thin filament in closer to the center. Then the head would let go, kind of go back to its original position, and grab actin again and bend again. So your myofibril is going to get shorter as a result of this. The myofilaments themselves do not get shorter, but they're moving past each other so that the whole myofilament gets shorter, and then when the whole myofibril gets shorter, and then when the whole myofibril gets shorter, that makes the whole muscle get shorter, usually. In order for that to happen, you have to have membrane potential changes in the muscle cell. You have to have action potentials in the muscle cell. So we can divide contraction into 4 different phases. First, excitation. That's when the nervous system is stimulating the muscle cell to contract. Then excitation-contraction coupling. That's basically an action potential spreading across the membrane of the muscle cell. Then you have contraction, which is when the thick and thin filaments actually move past each other. And then you have relaxation, when the filaments let go of each other and the muscle returns to its original length and the membrane potential resets. So we'll go through each of these phases in detail. First, I want to remind you about the membrane potential itself. It's composed of a separation of charge across the membrane, so that on one side you have more positive charges that would be outside of the cell, you have more positive charges, then inside of the cell you have more negative charges. And so right there at the membrane you have a difference in charge, which creates a membrane potential or a voltage across the membrane. In neurons, the resting membrane potential is -70 millivolts. In muscle cells, it is a little lower. It's -90 millivolts in skeletal muscle cells. And again, that's always measured as the inside compared to the outside. So if you have a negative membrane potential, that means that the inside is more negative than the outside. Overall, of course, you have electric neutrality, which means that you don't have a difference in how many positive and negative charges there are in the whole tissue. Just if you look at the membrane, you can see a difference right by the membrane. Okay. So you have different channels in the membrane of skeletal muscles that allow ions to cross the membrane and affect the membrane potential. You have leak channels that are randomly opening and closing. You have the ligand-gated channels that open when a ligand binds them, when a specific chemical binds them. And then you have voltage-gated channels that open when the membrane potential reaches a certain value. And then finally, you have the sodium-potassium pump, also called the sodium-potassium ATPase. That's not a channel, but it does move ions across the membrane. So the leak channels are involved in generating the resting membrane potential in muscle cells and in neurons. For muscle cells, most of your leak channels are for potassium. So you have always some potassium leaving the cell. As you remember, there's lots of potassium inside the cell and only a little bit outside. So if you open a channel for potassium, it will move down its concentration gradient from inside where you have a lot to outside where you have little, taking its positive charges with it. So anytime you have potassium leaving the cell, that makes the inside more negative and the outside more positive, so it causes the membrane potential to become more negative. You also have some sodium coming into the cell through the leak channels. Sodium is more concentrated outside of the cell than inside, so when channels open for sodium, sodium will enter a cell down its concentration gradient, and that means that positive charges would be entering the cell, which makes the membrane potential more positive. But overall, you have more potassium going through the leak channels than you have sodium. which means that overall the membrane potential will drop because of those leak channels. And that's part of what makes it negative 90 millivolts at rest. The sodium-potassium pump is also important in establishing the resting membrane potential, but it also just maintains the ion gradients, the concentration gradients for ions. That's probably its most important role. So the sodium-potassium ATPase uses ATP. That's why it's called an ATPase, because it breaks ATP down into the phosphate and ADP. And it uses the energy from doing that to pump 3 sodium ions outside of the cell and to bring 2 potassium ions into the cell. And there they are coming into the cell. And it's just continually cycling like that, putting sodium out and bringing potassium in. That costs ATP because you are moving the ions against their concentration gradient. You're moving the sodium to where you have more already and moving the potassium to where you have more of that already. Anytime you're going against a concentration gradient, it costs energy. So that's really important because if your concentration gradients for the ions change, you're not going to have them moving through channels when the channels open the way that they're supposed to. Then you wouldn't be able to have action potentials in the muscle cells. And if you can't have action potentials in the muscle cells or in the neurons, of course, then you won't have muscle contraction or the function of neurons. The last 2 types of channels are involved with the muscle contractions themselves. So you have the ligand-gated channels. Those are important for the excitation phase of muscle contraction. They would be opening in response to binding of a neurotransmitter. So you find those channels at the neuromuscular junction, and they respond to acetylcholine released by a motor neuron onto the muscle. Then you have the voltage-gated channels. Those are responsible for action potentials. So those are important for the excitation-contraction coupling phase of muscle contraction. And to review again, whenever the membrane potential is changing to become more positive, we call that depolarization. If it's changing to become more negative, we call that either repolarization or hyperpolarization. If the membrane potential is higher than rest and is going back down to the resting membrane potential, we call that repolarization. But if it's already at the resting membrane potential and then it's dropping even lower than that, we call that hyperpolarization. Alrighty, just to review again, action potentials versus local potentials. Action potentials are all or nothing, so they always look the same, and it's a strong signal. I guess there was actually a graph of one on the previous slide, where you have the depolarization phase that's caused by sodium entering the cell through voltage-gated sodium channels. Then your sodium channels close and voltage-gated potassium channels open. Potassium will begin leaving the cell. That causes the repolarization phase, and the membrane potential will drop back down. Then once these potassium channels close, once they start closing, they're kind of slow to close. So they'll be open for a while after they've started to close, and the membrane potential will continue to drop as potassium continues to leave as long as those channels are open, which gives you the hyperpolarization phase. So in your muscle cells, the action potential actually looks a little bit different. You don't have a hyperpolarization phase. You have a depolarization phase and a repolarization phase only. And that's because the membrane potential is lower at rest in your muscle cells than in neurons. So for a muscle cell action potential, you start at your resting membrane potential of negative 90 millivolts. Then something will happen to depolarize a cell to a threshold of negative 55 millivolts. That something would be a local potential. At negative 55 millivolts, the voltage-gated sodium channels open. Sodium rushes into the cell, so positive charges are entering the cell. That takes the membrane potential more positive, all the way up to the peak value of around 30 millivolts. At that point, the sodium channels close and your voltage-gated potassium channels open. Technically speaking, they had already started opening back whenever the sodium channels opened, but they're pretty slow to open, so they're barely actually opening now at peak depolarization. Then your potassium will start leaving the cell through those channels. That means that positive charges are leaving the cell. That means that the inside becomes more negative. Less positive is more negative. So that gives you the repolarization phase of the action potential. Around negative 55 millivolts, the potassium channels will be triggered to close again, but they're slow to close, so you'll still have potassium leaving the cell and the membrane potential dropping more and more negative until they actually finally close at the end of the repolarization phase. At that point, you're back at negative 90 millivolts. In neurons, the hyperpolarization phase goes down to negative 90 millivolts, but in muscles, since that's your resting membrane potential, when you go down to negative 90, that's not hyperpolarization, it's just repolarization. And I'll just add that the action potentials are self-regenerating, which means that they spread across the entire membrane. So if you have one segment of the membrane going through the depolarization phase of your action potential, the sodium ions will diffuse along the membrane and open new voltage-gated sodium channels, and then that section of the membrane will enter the depolarization phase of an action potential, and so it will propagate and spread across the entire membrane. So that's action potentials in muscle cells. Local potentials are membrane potential changes that do not spread, which is why they're local, and they are not always the same. They can be different sizes, different intensities, and different directions. So that means they can make the membrane potential become more positive or more negative. In neurons, you have lots of different local potentials. In muscles, you actually have just one type, the end plate potential, which you find at the neuromuscular junction. So at the neuromuscular junction, you would have acetylcholine released by a motor neuron that would activate ligand-gated ion channels, and then you'll have ions moving through those channels, which causes an end-plate potential, and then those end-plate potentials will kind of add up to take the membrane to threshold, and that starts an action potential. So local potentials are always adding up to start an action potential if they can raise the membrane potential to negative 55 millivolts or above. Okay, so here's more about the actual excitation phase. So the excitation phase focuses on activity at the neuromuscular junction, which would be the contact or the synapse between a motor neuron and a skeletal muscle cell. So your lower motor neurons are all sending their axons to a muscle cell. And then those axons will branch at the end, and you'll have several different neuromuscular junctions from that one motor neuron axon. At each of those neuromuscular junctions, it's similar to a regular synapse in how it operates. It's similar to a regular synapse in the nervous system, but it is a little bit different. So you still have the axon terminal, which is the ending of the motor neuron axon, and you have the synaptic cleft, which is the little space between the motor neuron and the muscle cell. But the membrane of the muscle cell at the neuromuscular junction is specialized. As you can see, it has these kind of folds in it. That's called the motor end plate. So this part of the membrane that has the folds where it's contacted by a motor neuron axon, that is the motor end plate. The neurotransmitter that's released, it would be stored in these vesicles in the axon terminal. It's always acetylcholine. So if you have activity at this neuromuscular junction, you're going to have acetylcholine released into the synaptic cleft. And then it's going to bind to its receptors, which are always nicotinic acetylcholine receptors that are abbreviated NACHR. The nicotinic acetylcholine receptor is a non-selective cation channel, which means that it will allow most any positively charged ion to cross through it, once it's opened. So when acetylcholine binds, it opens, and then positively charged ions can move through it. So that means that sodium can move into the cell, potassium can move out of the cell through that channel, and sometimes you can even have calcium going through the channel as well. So activity at the neuromuscular junction would be the excitation phase of skeletal muscle contraction. You have an action potential arriving at the motor neuron axon, at the axon terminal. When that action potential arrives, it depolarizes the membrane, and that activates the voltage-gated calcium channels at the axon terminal. Then calcium will enter the cell because you have more calcium outside of the cell and less inside of the cell. Once calcium gets into the axon terminal, it's going to bind to proteins on these synaptic vesicles and cause the vesicles to fuse with the axon terminal membrane, and that releases the neurotransmitter acetylcholine into the synaptic cleft. The acetylcholine is going to cross to the other side of the synaptic cleft and bind to its receptors, which are nicotinic acetylcholine receptors, a type of ligand-gated ion channel. Once the acetylcholine binds, the channel will open, you'll have potassium leaving and sodium entering, but actually sodium will be entering faster than potassium leaves. So although you have two ions moving through the channel, it's really the sodium coming in that is dominating the kind of effect on the membrane potential as a result of the opening of this channel. So sodium will be coming into the cell, and that will be taking the membrane potential more positive in an end-plate potential. So all these sodiums entering the cell create an end-plate potential, which is depolarizing. Then to clean up, you would actually have an enzyme, acetylcholinesterase, degrade the acetylcholine, which that enzyme is not shown here in this diagram, but it would be there. Okay, that is basically the excitation phase. Activity at the neuromuscular junction is the excitation phase. To go through those steps again, you have an action potential arriving at the axon terminal of the motor neuron that depolarizes the axon terminal and opens the voltage-gated calcium channels. Calcium enters the axon terminal. It causes the vesicles containing neurotransmitter to fuse with the membrane and release their neurotransmitter, which is acetylcholine. Then acetylcholine crosses the synaptic cleft and binds to its receptors, such as right here. Those receptors are nicotinic acetylcholine receptors. Once acetylcholine binds to those receptors, they will open and ions can cross. The main ion that crosses is sodium. You also have potassium exiting, but mostly you have sodium entering. So the sodium is going to enter faster than the potassium can exit. So all that sodium entering is going to cause an end plate potential, a depolarizing local potential called an end plate potential. So since the end plate potential is always caused by sodium entering the cell, entering the muscle cell, it's always depolarizing. Its size can be different strengths. How big it is depends on how much acetylcholine you had released and how many of these channels were open for how long. So these end plate potentials are going to be generated as long as you have action potentials arriving at the motor neuron. You're going to be making end plate potentials in the muscle cell. Those end plate potentials are going to spread as the sodium ions diffuse to either side, well, to all sides. It would actually be like 3D, like a circle. When they get to the regular sarcolemma, so when they get away from the motor end plate part of the membrane, then they will open voltage-gated sodium channels, and that kicks off an action potential, which would be the next phase of muscle contraction, excitation-contraction coupling. So the excitation phase begins when you have the action potential arriving at the motor neuron, and it ends when you have end plate potentials triggering an action potential. Then you also have acetylcholinesterase in here, which would be shown here and here, degrading the acetylcholine after it's been released to end the signal. So then the next phase is excitation-contraction coupling. Excitation-contraction coupling basically involves an action potential spreading across the entire sarcolemma of the muscle cell. So it would start at the neuromuscular junction, at the edges of the neuromuscular junction, so at the edges of the motor end plate, and it'll spread in all directions across the entire sarcolemma. It will also spread down into the T-tubules, because those are continuous with the sarcolemma. They're connected to the sarcolemma, and they have voltage-gated channels, voltage-gated sodium and potassium channels in them. So your action potential will not only spread across the sarcolemma, but down into all of the T-tubules. And at that point, the triads will come into play. So more detail on those action potentials in myocytes. In the beginning, to start one of these action potentials, you have to have the end plate potentials. Those are abbreviated as EPP there in the figure. End plate potentials adding up to threshold to take the membrane potential from rest at -90 to threshold at -55. At -55 millivolts, these voltage-gated sodium channels will open, and sodium will rush in. That starts the depolarization phase of the action potential. So the membrane potential will shoot up all the way to about 30 millivolts. Meanwhile, your potassium channels were triggered to open back at -55 millivolts, but they're very slow to open. So by the time you're at 30 millivolts, they are finally actually opening, and potassium will start entering the cell even as the sodium channels close. Of course, the sodium channels are not going to just close, they lock. Their lock gate swings into place, and they are locked. The potassium channels will actually be open, and potassium will be leaving the cell. When potassium leaves the cell, you are losing positive charges from the inside, so that means the inside is becoming more negative, so the membrane potential will start dropping, and that is the repolarization phase. Once you get back down to around -55 millivolts, the potassium channels will be triggered to close, but again, they're very slow to close. So you're still going to have them being open and potassium leaving the cell all the way until you get down to -90 millivolts. At that point, the potassium channels will actually close, and your sodium channels will have already reset. That is going to spread across the entire membrane, because once you have the first part of the membrane right by the motor end plate enter the depolarization phase, you'll have sodium coming in. It will diffuse along the membrane and open new voltage-gated sodium channels a little bit further distant on the membrane, and that starts an action potential in that part of the membrane, and so on and so forth. So your action potential will be spreading across the entire membrane of the muscle cell, across the entire sarcolemma. It will also spread down into the T-tubules. They also have voltage-gated channels for sodium and potassium, so they can have action potentials. Another thing you have in the triads is these voltage-gated calcium channels. The voltage-gated calcium channels are actually embedded in the membrane of the terminal cisternae of the sarcoplasmic reticulum. That would be the thickened part of the sarcoplasmic reticulum that is right next to the T-tubules on either side. So those calcium channels, the voltage-gated calcium channels, are in the sarcolemma membrane, in the membrane of the terminal cisternae. But they also have this little linker protein that is embedded in the membrane of the T-tubule. And it's that linker protein that is actually voltage sensitive. So normally it's kind of relaxed and the channel will be closed, but if you have an action potential go down the T-tubule, then you're going to have depolarization going all along the T-tubule, and that causes this linker protein to change its shape. The coil of it gets tighter, so it shortens. When it shortens like that, that pulls the little, flap on the calcium channel open, so that now the calcium channel is open. So it's a little bit strange because the channel is in the terminal cisterna membrane, but it responds to voltage changes in the T-tubule membrane because of this linker protein. Once this voltage-gated calcium channel is open, then you're going to have calcium that was stored in the sarcoplasmic reticulum, leaving the sarcoplasmic reticulum down its concentration gradient, moving into the cytosol of the cell. And that is the end of the excitation-contraction coupling phase. It starts whenever you have an action potential generated, then the action potential will spread across the entire sarcolemma and down all of the T-tubules, and then that phase will end when you have the voltage-gated calcium channels in the terminal cisternae opening and calcium leaving the sarcoplasmic reticulum. So here's a summary showing all of that. So here you have the excitation phase at the neuromuscular junction, and then here you have the excitation-contraction coupling phase. So once your acetylcholine is released, it activates the nicotinic acetylcholine receptors, sodium enters the muscle cell, that causes an end plate potential. That end plate potential will spread to to all sides of the motor end plate. When these sodium ions diffuse to the membrane next to the motor end plate that has voltage-gated channels in it, they're going to open the voltage-gated sodium channels. Then you have an action potential starting there, sodium entering, those ions will diffuse down and open the next voltage-gated sodium channel. So then this part of the membrane will enter the action potential depolarization phase. Sodium will be coming in, it'll diffuse along the membrane down to the next voltage-gated sodium channel and open that, and so the action potential will spread across the whole sarcolemma membrane in all directions in 360 degrees and down all of these T-tubules. Once it gets down into the T-tubules, it will cause the depolarization, will cause this linker protein to shorten, and that pulls open the voltage-gated calcium channels in the terminal cisternae membrane, and then calcium will exit the sarcoplasmic reticulum into the cytosol. And that's the end of the excitation-contraction coupling phase. Okay, so just to summarize some of that and compare it to what we learned about with neurons. So your local potentials in neurons are carried by all types of ions, usually sodium, potassium, chloride, or calcium. In myocytes, they're only carried by sodium. In neurons, they can be depolarizing or hyperpolarizing. If they're depolarizing, we call them EPSPs, or excitatory postsynaptic potentials. If they're hyperpolarizing, we call them IPSPs, or inhibitory postsynaptic potentials. But in myocytes, we only have one type of local potential carried by the sodium ion. It's always depolarizing, and we call it the end plate potential, or EPP. In neurons, we find local potentials in the dendrites and the cell body. In the myocytes, we only find them at the motor end plates, right at the neuromuscular junction. In both cases, in neurons and in myocytes, those local potentials are triggered by a neurotransmitter. In neurons, it could be lots of different neurotransmitters. In muscles, it's only acetylcholine that triggers the local potentials. And in your neurons, the local potentials would be traveling down the dendrites and across the cell body to the axon hillock. And at the axon hillock, they add up together to see whether or not they will be able to initiate an action potential. In the myocytes, your local potentials will spread to the ends of the motor end plate and sum up there to see whether they will initiate an action potential. Action potentials are also a bit different between neurons and myocytes. In neurons, they have depolarization phase, the repolarization phase, and the hyperpolarization phase. In myocytes, they only have the depolarization and repolarization phases, and that's again because the resting membrane potential is -90 millivolts in myocytes, not -70 like it is in neurons. In both cases, they use the same channels, voltage-gated sodium and voltage-gated potassium channels. Voltage-gated sodium for depolarization, voltage-gated potassium for repolarization, and for hyperpolarization in neurons. Since they're action potentials, they are always the same size, they always have the same characteristics. In neurons, they're going to propagate along the axon down to the axon terminals. In myocytes, they'll propagate along the entire sarcolemma in all directions and down into the T-tubules. And in both cases, once they get to where they're going, they cause the release of calcium. And neurons, once the action potential reaches the axon terminal, it opens the voltage-gated calcium channels. In myocytes, once the action potential reaches the T-tubules, it opens the voltage-gated calcium channels that are in the terminal cisternae, and that releases calcium from the sarcoplasmic reticulum. And we also have some membrane potential differences. In neurons, negative 70 millivolts is the resting membrane potential. In myocytes, it's negative 90. The threshold is always the same, negative 55 millivolts. The threshold is determined by the voltage-gated sodium channel itself, not by anything else. You have the peak depolarization of 30 millivolts in an action potential. That's the same for both neurons and myocytes. The minimum potential reached during an action potential is negative 90 for both cell types, but negative 90 in neurons is below the resting membrane potential, so that's hyperpolarization, whereas in myocytes, negative 90 is the resting membrane potential, so that's just the end of repolarization. The ion concentrations are basically the same in both types of cells. You always have sodium and chloride and calcium in a high concentration outside of the cell, low concentration inside of the cell, and then potassium always has a high concentration inside of the cell and a low concentration outside of the cell. But in myocytes, you also have calcium stored inside the cell in the sarcoplasmic reticulum. So the cytosol of the muscle cell has a low concentration of calcium, and then inside the sarcoplasmic reticulum, you have a high concentration of calcium. And the resting membrane potential in both cases is created by the interaction of the sodium-potassium pump and the leak channels. It's just that the leak channels operate a little differently between the neurons and the myocytes, which makes the membrane potential in the myocytes a little lower at rest than it is in neurons. Okay, so next we would have the contraction phase. Before we can talk about the contraction phase, we need to talk about what the myofilaments actually look like at rest and when that changes in the contraction phase. So for your thick myofilaments, as you remember, they're made mostly of myosin, and those myosin heads are found at either end of the thick filament, and the head can bend at the hinge down in towards the tail. Then your thin filaments are mostly made out of actin. The active site on the actin is normally blocked, or at rest it is blocked, by tropomyosin. And then the tropomyosin is anchored in place by troponin. So in order to have contraction, you have to reveal the actin active sites so that myosin heads can grab them. In order for that to happen, you have to move the tropomyosin out of the way. In order to move tropomyosin, you have to remove troponin, which is anchoring it in place. So after you have the excitation-contraction coupling phase, at the very end of that you get calcium entering, calcium entering the sarcoplasmic reticulum. Calcium exiting the sarcoplasmic reticulum, entering the cytosol. When the calcium enters the cytosol, it's going to bind to troponin. So here this troponin has a couple little green calciums attached to it. When calcium binds to troponin, it's going to move it out of the way. And when the troponin moves, then the tropomyosin is going to move out of the way. So now these actin active sites are exposed. So normally they are covered at rest, and in the presence of calcium, troponin moves, then tropomyosin moves, and then your actin active sites are exposed. Once those active sites are exposed, contraction can begin. Once those actin active sites are exposed, then the myosin head can grab the actin active site, and that is called a crossbridge. An actual attachment between the myosin head and the actin active site is a crossbridge. So at the beginning of your contraction phase, crossbridges are going to form between myosin and actin. So once you have the crossbridge formed between the myosin head and the actin active site, then you can begin cross-bridge cycling. So we would be starting right here with our myosin head cross-bridged with our actin active site. Then the myosin head will bend. That's called the power stroke. When the myosin head bends, it's going to pull the thin filament in towards the center of the thick filament. Then the myosin head is going to use ATP to detach, to let go of the actin active site. It'll use ATP again to reset or cock to its original position. And then it will grab the actin active site again, and the cycle will repeat. You'll have another power stroke, use ATP for myosin to let go of actin, use another ATP to cock myosin, then myosin grabs the actin active site, power stroke, so on and so forth, in a cycle that will repeat as long as the muscle is contracting. So as cross-bridge cycling continues, the thin filaments are moving closer and closer in towards the center of the thick filaments. The myofibrils are getting shorter, and the myocytes themselves are getting shorter, which would generally mean that the entire muscle is getting shorter. There's a limit to how long cross-bridge cycling can go. As it goes, you are going to be moving the thin filaments in towards the center of the thick filaments. You have the M line there, which is just made of proteins linking the different thick filaments together. So as you cross-bridge cycle, the thin filament moves closer in towards the M line, towards the center of the thick filaments. Then the myosin is going to grab a new actin head and do the power stroke and bring that closer into the center, and then it would grab a new actin active site and do the same. So eventually these thin filaments are going to actually reach the center of the thick filament, and then they're not able to move anymore because they're blocked by the proteins that are there. So at that point, cross-bridge cycling kind of has to stop. So there's always that limit on how much the myofibril can shorten and how much the whole muscle can shorten. Then the final phase of muscle contraction is relaxation, which is important because if you didn't do that, then your muscles would kind of just be paralyzed in the contracted state. And the relaxation phase begins as soon as you have no more action potentials arriving at the neuromuscular junction. So as soon as this motor neuron stops sending action potentials, then the relaxation phase will begin. First, you're going to have any acetylcholine that is left in the synaptic cleft degraded by acetylcholinesterase. Then your final action potential that had gone out will enter the repolarization phase, and the membrane potential will reset to rest. That repolarization will spread down into the T-tubules. Your voltage-gated calcium channels in the terminal cisternae are going to close, so you stop releasing more calcium from the sarcoplasmic reticulum. And then you're going to have these calcium pumps in the terminal cisternae taking calcium, using ATP to take calcium from the cytosol and pump it back into the sarcoplasmic reticulum. So that costs ATP because you're moving calcium from where you have less to where you have more, going up its concentration gradient. As the calcium concentration in the cytosol drops, you won't have enough calcium left for it to be bound to troponin. So calcium will let go of the troponin, and then when that happens, troponin is going to move back into its original position, and when it does that, it pushes tropomyosin back into its original position so that the actin active sites will be covered up. Once those active sites are covered up, you can't have any more cross-bridge cycling. And then the whole muscle will relax as the thick filaments detach from the thin filaments. The myofibrils will go back to their original length. The muscle cells will go back to their original length, and the whole muscle will go back to its original length. So just to summarize all of that again, the first phase of muscle contraction is excitation. That is activity at the neuromuscular junction. You have your action potential arrive down that axon of the motor neuron. It opens a voltage-gated calcium channel, which isn't shown here, but it is there. Then calcium would enter into the axon terminal. It would cause these vesicles containing acetylcholine to fuse with the membrane and release acetylcholine. Acetylcholine will bind to nicotinic acetylcholine receptors and open them. Then you're going to have sodium entering through those receptors. That causes depolarization, which we call an EPP, or end plate potential. Those sodium ions will diffuse away from the motor end plate to start an action potential, and that is the end of the excitation phase. Then as soon as you have that action potential begin, that's the excitation-contraction coupling phase. So the sodium ions will add up to enough depolarization to bring this membrane from negative 90 millivolts to negative 55. And then the voltage-gated sodium channels there will open. Sodium will rush in the depolarization phase of an action potential. That action potential will propagate along the entire sarcolemma and down into the T-tubules. When it gets into the T-tubules, it's going to depolarize the membrane there, and that causes this linker protein to shorten its coil, and that will pull open the voltage-gated calcium channels that are in the terminal cisternae. And then calcium will exit the sarcoplasmic reticulum down its concentration gradient into the cytosol. So all of that is excitation-contraction coupling. From the beginning of the action potential to the release of calcium from the sarcoplasmic reticulum is excitation-contraction coupling. Then you would have the contraction phase of muscle contraction, in which your calcium binds to troponin. That makes troponin let go of tropomyosin, and tropomyosin is going to move or slide away from the actin active sites. So now you have those active sites exposed. Then the myosin head will grab an actin active site, and you'll begin crossbridge cycling. The myosin will undergo the power stroke, pulling the thin filament in towards the center of the thick filament. It will use ATP to let go of the thin filament. It will use ATP again to recock. Moving back to its original position, it will grab an actin active site again, a different one this time. It will go through the power stroke again, so on and so forth. So that's your crossbridge cycling. That is the contraction phase of muscle contraction, and that will continue as long as you still have calcium in the cytosol of the muscle cell. The final phase of muscle contraction is relaxation. That begins as soon as you have action potentials stop arriving at the motor neuron's axon terminal. So when those action potentials stop, then you'll have acetylcholinesterase degrade the remaining acetylcholine in the cleft. The last action potential will finish up with its repolarization phase, so the membrane potential will return to the resting value. That will spread down the T-tubules as well, everything returning to resting values. These voltage-gated calcium channels will close, and then the calcium pumps will start using ATP to pump calcium back into the sarcoplasmic reticulum. And once the calcium concentration in the cytosol gets low enough, then calcium will let go of troponin. Troponin will move back into its original location and push tropomyosin back into its original location as well, blocking the actin active sites, which prevents crossbridge cycling from continuing. So that's the very end of muscle contraction. Okay, so that's the physiology of muscle contraction. Now we'll talk about where muscle gets its energy from, because this process requires a lot of energy. Okay, so first we'll talk about where you're actually using energy in muscle contraction. So first you need energy for the myosin heads to grab actin in the first place. That's because in order to grab actin, they have to be cocked. It takes energy for them, or it takes ATP for them to cock. So they use ATP to cock, then they can grab actin. Then they also need to use ATP again to let go of actin. So they actually need to use it twice. It also costs ATP to put the calcium back in the sarcoplasmic reticulum. The calcium pump is also called the calcium ATPase because it uses ATP. And then finally, it takes ATP to keep the ion concentration gradients constant between the inside and the outside of the cell. So keeping the outside of the cell high in sodium while keeping the inside of the cell high in potassium requires ATP. That's used by the sodium-potassium ATPase or the sodium-potassium pump. So you have ATP being used twice during actual crossbridge cycling, and you have it being used to keep your ions where they need to be so they will have the right concentration gradients. Sodium outside of the cell, potassium inside of the cell, and calcium inside the sarcoplasmic reticulum. Okay, so muscles have a lot of sources they can use for ATP. First, they will just use whatever ATP they have lying around, which is usually around 3 seconds worth, or it's enough to power the muscle cells to contract for about 3 seconds. After that, the muscle cells start having to make ATP to contract for longer. Their next source of ATP is creatine phosphate, which will power the contraction for about 10 more seconds. Or give enough ATP to power the contraction for about 10 more seconds. Creatine phosphate is found in the cytosol of muscle cells, and you have an enzyme there, creatine kinase, which can combine creatine phosphate with ADP, which is the spent form of ATP, to give you creatine and ATP. So basically what that enzyme is doing is taking the phosphate group from creatine phosphate and giving it to the ADP, that's adenosine diphosphate, and that makes ATP, or adenosine triphosphate. So you're just transferring a phosphate group from creatine to ATP. That gives you just creatine left over from that. So with that creatine, you can either later reattach another phosphate group to it, so you can do this again, or you can convert it to creatinine, which would be eliminated as a waste product by the kidneys. So that's another source of ATP. Then after that runs out, the cell will turn to glycolysis, although actually it will just always be doing glycolysis actually. But glycolysis will give enough ATP to power the muscle contraction for about 35 seconds. Glycolysis is a type of anaerobic catabolism, which means that it's a way of breaking down nutrients to get energy out of them without using oxygen. It occurs in the cytosol of the cell. Actually, every cell in your body does glycolysis for energy, but muscle cells need an awful lot of energy, so they will be doing quite a lot of glycolysis if you're using them. So glycolysis, where's my pointer? It's actually shown on the side here. So as you can see, this is a multi-step process. This is actually just a summary. There's actually a bunch of steps that aren't even shown here. So all these arrows, they don't have anything. That's actually a step that they're not showing. You're starting with glucose, and you're converting glucose into pyruvate, ultimately through this series of many steps, which you definitely don't need to know all these steps. But glucose has 6 carbons in it. At this step right here, you are splitting the 6 carbons into 2, so you get 2 molecules that each have 3 carbons, and then those are converted finally to pyruvate, and pyruvate has 3 carbons. So each glucose is going to give you 2 pyruvates. So you start with glucose and you get 2 pyruvate. This process actually does require ATP at the beginning. The ATP is used to add phosphates to the glucose, which gives you this fructose 1,6-bisphosphate, which you don't need to know, but FYI, that's why they're being used. But at the end, once you're making your pyruvate, that's when you start making ATP out of this process. So you use 2 ATP, but then you get 4 ATP. 2 ATP for every pyruvate that you make, and you make 2 pyruvates, so it gives you 4 ATP total that you're making from this process. And then the last thing is that you are using NAD+. NAD+ is an electron carrier, as you may remember from Intro to Biology. So when you add electrons to NAD+, it becomes NADH, and it can then carry those electrons elsewhere in the cell so they can be used for something. So from this whole glycolysis process, you're taking glucose, converting it into 2 pyruvates. You are using 2 ATP, you're making 4 ATP, and you're also making 2 NADHs. You're adding electrons to 2 NAD pluses to give you 2 NADHs. The glucose that you're using for this process can come from multiple places. Of course, you have glucose in the blood. If you're kind of working out, then your body is going to boost the amount of glucose in your blood using the sympathetic nervous system and different hormones. So that will provide extra glucose for the muscles to do glycolysis. But you can also have glucose stored in the muscles from glycogen. Stored in the muscles as glycogen. The liver is the main source of stored glycogen in the body, but muscles can also store some glucose as glycogen and use it when they start contracting. In order for glycolysis to continue, you need, of course, to always have more glucose for it. You also need to have more NAD+. So once you've made this NADH, you have to turn that over back into NAD+ in order to continue doing glycolysis. And some of the next types of metabolism we'll talk about are where you're using that NADH to get NAD+ back. Then for this pyruvate, you can either use that for aerobic catabolism. Well, you would actually be using NADH for aerobic catabolism as well. That is using nutrients, breaking down nutrients to get energy in a way that does require oxygen. So that's one fate for the pyruvate. Alternatively, you could ferment this pyruvate to make lactic acid. If you're doing the fermentation, then you're going to be using the pyruvate and the NADH. You'll end up getting NAD+ and lactic acid or lactate. Then the lactate would go to the liver to be converted back to glucose, or it can go on and substitute for pyruvate in the next steps of metabolism. Okay, once the muscle cell runs out of ATP from other sources, it's going to turn to oxidative catabolism, which is also called aerobic respiration or aerobic catabolism or cellular respiration. All of those different names just mean that it's breaking down nutrients to get energy, and it uses oxygen. Oxidative catabolism can supply ATP indefinitely. As long as you have a supply of oxygen, it can make enough ATP to fuel the contractions because it makes a very, very large amount of ATP. Actually, of course, your muscle cells are always doing glycolysis. They're always doing oxidative catabolism. But if they start contracting, then they're going to do more of it to get more ATP. Oxidative catabolism occurs in the mitochondria. Right here. It would be occurring one of the major processes is actually occurring in the membrane here, in the little internal membrane of the mitochondria, and then also you have some occurring in the matrix of the mitochondria. There's a lot of different steps in oxidative catabolism. Really, very, very, very many steps. And you can use a lot of different starting materials. Usually cells would be using pyruvate that comes from glycolysis. But you could also be using fatty acids from fats or amino acids from proteins. So if you are kind of running low on glucose, then you can't make pyruvate in glycolysis. That would be like if you're kind of starving, and then your body would switch to surviving off of fat or protein. What that means is that it would be breaking down the fats or the proteins to make fatty acids or amino acids, and then using those things to make ATP in oxidative catabolism instead of glucose. So in addition to that starting material, the pyruvate or fatty acid or amino acid, you would be using oxygen as well. Of course, the oxygen is coming from the blood, but you also have oxygen that's stored in muscle cells on myoglobin. Myoglobin is like, it's very similar to hemoglobin. Hemoglobin is the protein inside your red blood cells that lets them carry oxygen, so it binds oxygen. Myoglobin is a very similar protein, but it's found in muscle cells, hence the myo in the name. And it just binds oxygen and stores it. So your muscles will always have a store of oxygen ready for them. because their oxygen needs are going to go up a lot if they start contracting and they need to do a lot more oxidative catabolism. You've got three basic processes in oxidative catabolism. You have pyruvate oxidation, the Krebs cycle. The Krebs cycle is also called the CCA or TCA cycle, the citric acid cycle, or the tricarboxylic acid cycle, but I like to call it Krebs cycle because it's shorter. And then finally you have oxidative phosphorylation as the last process. So first you start with pyruvate oxidation. That is the simplest process in oxidative catabolism. Pyruvate oxidation starts with pyruvate and NAD+, and coenzyme A or CoA. And it's going to be transferring two of the carbons from the pyruvate onto the CoA, which gives you acetyl-CoA. And it also transfers electrons onto the NAD+, which gives NADH, and then the last carbon from pyruvate ends up as carbon dioxide. So here you have a graphic of that process where you're starting with pyruvate that has 3 carbons. You end up with 2 of those carbons on acetyl-CoA. So the CoA attaches to 2 of those carbons to make acetyl-CoA. Then your last carbon ends up as carbon dioxide, which is a waste. And then you're also adding electrons to NAD+, which gives you NADH. So glycolysis made NADH, and pyruvate oxidation also makes more NADH. And you're going to have to do something with that NADH later on. Then the next process is the Krebs cycle, which is shown right here. This again is like only part of the Krebs cycle. The Krebs cycle has a bunch of different steps in it, and a lot of them are omitted in this diagram actually. In the Krebs cycle, you start with oxaloacetate. You're adding the carbons from acetyl-CoA to that oxaloacetate, which gives you citrate. That's why it's called a citric acid cycle sometimes. And then you're just going to go around the cycle modifying the citrate at each turn until you have converted it back to oxaloacetate. And at that point, the cycle can repeat. So it's a cycle because you start with oxaloacetate and you also end with oxaloacetate, and you can keep going around in a circle. As you're going around the circle, you're going to end up removing some of these carbons, and they will be lost as carbon dioxide as a waste product. You're also going to end up adding electrons to NAD+ to give you NADH at 3 different points. You're going to add electrons to FAD to give you FADH2. FAD is another electron carrier, basically like NAD+. And finally, you're going to be adding phosphates to ADP to make ATP. In this graphic, it shows GTP. You can make GTP or ATP in this way, but for our purposes, we'll just remember that you make ATP. Ultimately, for every turn of this cycle, you would be making 3 NADHs, 1 FADH2, and 1 ATP. But actually, remember that since you get 2 pyruvates out of glycolysis, that means that for every glucose, you would do 2 pyruvate oxidations and 2 turns of the Krebs cycle. So you would be getting 2 ATP total for each glucose out of the Krebs cycle. Then finally, we have the last process in oxidative catabolism, which is oxidative phosphorylation. In oxidative phosphorylation, you're finally going to use that NADH and the FADH2 that you made in glycolysis, in pyruvate oxidation, and in the Krebs cycle. And this is also the step that uses oxygen. This is the step that is occurring in the membrane, in the internal membrane of the mitochondria. You have 2 kind of parts of oxidative phosphorylation. First, you have this is the electron transport chain, this part here, and then second you have chemiosmosis, which is the last part here. So in the first part, what's happening is that you are taking your NADH and your FADH2, they are losing their electrons to give you NAD+ and FAD. So this is how you get back NAD+ and FAD. These electrons are going to move onto these proteins that are embedded in the membrane, in the inner membrane of the mitochondrion. And the electrons are going to be transferred from one protein to the next. as they move from one protein to the next, they are losing energy. The energy that they lose is used to pump hydrogen ions across that membrane into this little space between the inner and outer membranes of the mitochondria. So you're building up hydrogen ions in this space as the process continues. Then as an electron reaches the end, as it gets to the last protein in the membrane, is going to be added to oxygen to make water. So that's why you need oxygen. You need something to take those electrons. It's called the final electron acceptor because once the electrons have gone through this electron transport chain, once they've moved across every protein in the chain, then they're added to the final electron acceptor to oxygen. Oxygen will accept them at the end, and that gives you water. And this is actually the reason why you need to breathe oxygen and why you will die without it. If you don't have oxygen here, then you don't have anything to take the electrons. The electrons will get stuck on these proteins. The whole thing will back up with electrons. NADH will come to drop off electrons, but it can't. There's nowhere to drop them off because there's already electrons here, and it will end up staying as NADH. That means you're not going to be making ATP from oxidative catabolism. The whole thing will shut down. It also means you're not going to be making ATP from glycolysis because you cannot do glycolysis if you're not getting this NAD+ back. So at this point we haven't made ATP yet. We've just made a concentration gradient for hydrogen ions, where you have a lot of hydrogen ions in this little space between the two membranes and only a little bit of hydrogen ions in the innermost space of the mitochondria. The next process, chemiosmosis, is where you actually make the ATP. That uses this protein here, which is called ATP synthase. This protein will allow hydrogen ions to flow down their concentration gradient, and it uses the energy of their motion to add a phosphate group to ADP, which gives you ATP. So this pump is, well, it's not exactly a pump actually. This ATP synthase is allowing hydrogen ions to flow into the innermost space of the mitochondrion, down the concentration gradient, and as they move, the energy is used to make ATP. Overall, how much ATP you can make this way kind of varies on how efficiently the process is occurring, but for each glucose you would make around 28 ATPs. In oxidative catabolism as a whole, 2 of those would have come from the Krebs cycle, and the remaining 26 would have come from ATP synthase in chemiosmosis. So you don't need to know the details of that exactly. You need to understand basically that these electrons are coming from NADH and FADH2, that they're moving from protein to protein on this chain here, and then they end up on oxygen, and as they move, hydrogen ions are pumped across the membrane, which creates a concentration gradient for hydrogen ions that is later used by ATP synthase to make ATP. A lot of ATP. So a summary of the overall energy sources for muscle, in addition to ATP that happens to be lying around, you have the creatine phosphate, which can make 1 ATP per creatine phosphate, about 10 seconds worth of ATP. And then you have glycolysis, which makes 2 ATP for every glucose. It also makes pyruvate, which could be converted to lactate, and it makes NADH. Then the pyruvate would go on to oxidative catabolism, which makes 28 ATP per glucose. First you have pyruvate oxidation, which does not make ATP, but it does convert pyruvate into acetyl-CoA so that it can enter Krebs. Then in the Krebs cycle, you're making 2 ATP, and you're also making a bunch of NADH and FADH2. Then in oxidative phosphorylation, that's where you're using NADH and FADH2 and oxygen, and that includes the NADH that came from glycolysis. And this oxidative phosphorylation is actually making the bulk of the ATP, 26 ATP. In this kind of graphic format here, here you have glucose entering glycolysis. It comes out as pyruvate, which would go through pyruvate oxidation to convert it to acetyl-CoA. Acetyl-CoA then enters the Krebs cycle, which makes a bunch of FADH, well, a bunch of NADH and some FADH2. Those are electron carriers that go to the electron transport chain of oxidative phosphorylation. You also have more NADH made from glycolysis. That's an electron carrier that enters the electron transport chain. And when you convert pyruvate to acetyl-CoA, you also get more NADH, and that enters the electron transport chain as an electron carrier. So all of those electron carriers are giving up their electrons to the proteins of the electron transport chain, ultimately transferring them to oxygen, which makes water. And then that builds up a hydrogen ion gradient that is used by ATP synthase to make ATP. You're also getting a little ATP out of glycolysis itself and a little ATP out of Krebs cycle, and you're making carbon dioxide from the pyruvate oxidation and from Krebs cycle. All that carbon dioxide is a waste product. This is another kind of summary of the process. You start with glucose and convert it to pyruvate in glycolysis. Then in pyruvate oxidation, you convert the pyruvate to acetyl-CoA. You also give off a carbon dioxide there and make an NADH. You would also make an NADH in glycolysis and ATP. Then that acetyl-CoA is going to enter the Krebs cycle, also called citric acid cycle. That generates NADH, FADH2, and ATP. Ultimately, the electrons on the NADH and FADH2 are going to enter oxidative phosphorylation at the electron transport chain. They will be used to create a hydrogen ion gradient that is then used by ATP synthase to make ATP. And you would also have more carbon dioxide coming off of the Krebs cycle. If you have something else like fatty acids or amino acids that you need to use instead of glucose, the amino acids can enter at different points. They could enter the Krebs cycle, they could enter pyruvate oxidation. It depends on which amino acid you're talking about. If you're breaking down fat, you're actually going to get fatty acids and glycerol from that. The glycerol is going to enter glycolysis. The fatty acids would enter the Krebs cycle as acetyl-CoA. But that you actually don't need to know. So you have this oxidative catabolism and glycolysis continuing at all times to provide the energy needs for your cells, for all of your cells actually. But when your muscles start contracting, then their ATP needs go up by a lot. So you're going to increase the rate at which you're doing glycolysis and oxidative catabolism, and you're going to start using that creatine phosphate to make ATP. So when you start contracting your muscles, you are going to be activating the sympathetic nervous system. That nervous system is going to be increasing the supply of oxygen and glucose to your muscles. It causes your liver to start releasing glucose from its stored glycogen. It's going to cause your fat tissue to start breaking down and giving supplies for the oxidative catabolism, glycerol and fatty acids. It's gonna cause your respiration rate to increase, your breathing rate to increase, so you're breathing in more oxygen. It also causes your heart rate to go up, the contraction force in the heart to go up. That moves blood faster through the system, and it's going to cause your blood vessels serving the muscles to dilate while other blood vessels constrict, so that your muscles will get a better blood supply. So all of those things are going to increase the supply of oxygen and or glucose to your muscles so that they can do more glycolysis and more oxidative catabolism. If you run out of oxygen or glucose, these processes will come crashing to a halt, and you will not be able to make enough ATP to fuel the muscle contractions, and the muscle will be forced to relax. Muscle fatigue occurs when your muscles are not able to produce as much force as they originally could because you have been using them too much, and they basically ran out of well, it's caused by different things, but one of the things that can cause it is that they run out of energy sources. Muscle fatigue is not the same as soreness. So when your muscles are fatigued, that means that you cannot make them contract the way you want. It doesn't mean that it hurts to contract them. Muscle soreness is when your muscles ache and hurt. Soreness usually occurs like you kind of notice it the next day. Muscle fatigue, you would be noticing it right away. We used to think that lactic acid buildup is what causes muscle soreness or muscle fatigue. It seems that that's not actually the case. It's more likely that the soreness is caused by damage to your muscles as you're using them. Little microscopic damage to the muscles or to the myofibrils. Fatigue has a number of causes, many different causes. So first, your motor neurons might not be able to send action potentials fast enough to make sure that you have a smooth muscle contraction. That would happen if you're not able to recycle the acetylcholine fast enough, or if you're not able to reset the ion concentration gradients fast enough. If you run out of ATP, of course, that would cause muscle fatigue. So if you don't have enough glucose or enough oxygen or enough creatine phosphate or enough glycogen or enough FAD and NAD+, if you have not enough of any one of those things, then you may have muscle fatigue. As your muscles are contracting, waste products will be building up. And some of those waste products can interfere with muscle contraction, causing muscle fatigue. So when you are using your ATP for muscle contractions, that gives you a free phosphate group. That free phosphate will bind to calcium, which prevents the calcium from binding troponin, and can end up having it can end up causing actin-active sites to be hidden. Also, ADP will build up as you're doing more muscle contractions. You're using more ATP, and that gives you ADP. And the ADP inhibits the calcium pump, so that you can't reset your calcium gradients as much. And then finally, as you're doing all of this metabolism in the mitochondria to make ATP, you're going to be generating reactive oxygen species, or ROS. Basically, whenever you're adding electrons to oxygen, that's supposed to make water, but sometimes it makes something different, something dangerous like hydrogen peroxide or another reactive oxygen species. Those things are very reactive. They'll react with pretty much anything. If they react with proteins that you need for the muscle contractions, then it will interfere with the muscle's ability to contract. Also, as you're continuing the muscle contractions, your ion concentrations will get out of whack. Right, you're going to end up with more sodium inside the cell, more potassium outside the cell. If the sodium-potassium pump can't reset those gradients fast enough, that can interfere with the action potentials that you need for muscle contraction. Also, as your ATP levels are dropping, the sodium-potassium pump is not going to work as well because it needs ATP to work. And then finally, different environmental conditions can cause fatigue. For instance, heat. For some reason, your muscles will become fatigued more quickly if you're using them in a hot environment. And this graph right here is just kind of showing you the muscle fatigue. So these would be muscle contractions. If you're trying to do something, you would have multiple contractions of the muscle. They should all kind of overlap with each other so that the muscle as a whole remains contracted, but individual cells would be contracting or not contracting depending on whether or not they're receiving action potentials. And then this is your basic amount of strength that you can produce with the muscle. And as you contract more and more, the amount of strength that you can produce starts to drop until you have fatigue. Okay, so now we'll talk about force produced by the muscles. So sort of the basic unit of muscle contraction is the twitch. The twitch is the amount of contraction in a single muscle cell that's produced by a single action potential in that cell. In order for the whole muscle to contract, you would need multiple twitches to occur, and they would have to occur quickly so they overlap with each other and end up adding up to be a force that will actually kind of mean something. If you just had a single twitch, you probably wouldn't even notice that it happened because it's so small. This is kind of a graph representing a twitch. You have 3 periods to a twitch: a latent period, a contraction period, and a relaxation period. In the latent period, that's the period between when you have signaled the muscle to contract and when the muscle actually starts to contract, or the muscle cell actually starts to contract. So first you have a stimulus, that would be an action potential coming along the motor neuron axon with activity at the neuromuscular junction, so that is the excitation phase of muscle contraction. Then you have an action potential generated that spreads across the whole sarcolemma and down into the T-tubules, so that is the excitation-contraction coupling phase. So in the latent period of a twitch, you have both the excitation phase and the excitation-contraction coupling phase of muscle contraction. Then in the contraction period of your twitch, you have calcium being released from the sarcoplasmic reticulum, and that's causing troponin to move out of the way and tropomyosin to move out of the way so that your actin active sites are revealed. Then you begin crossbridge cycling. As crossbridge cycling continues, the amount of force that you're generating from this muscle cell increases as the myofibrils get shorter and shorter. So this would be corresponding to the contraction phase of muscle contraction. Then at some point you're going to have the relaxation phase, or the relaxation period. So once you have reset the membrane potential from that action potential on the muscle cell, you're going to then start pumping calcium back into the sarcoplasmic reticulum. As calcium is pumped back into the sarcoplasmic reticulum, some of it will let go of troponin. That's going to end up having the actin active site hidden. So you're going to have fewer cross bridges between myosin and actin. As the calcium levels drop, you have fewer and fewer cross bridges and less force generated by that muscle cell. So this would be corresponding to the relaxation phase of muscle contraction. So that individual twitch is very small. It's not enough to actually get work done. In order to do meaningful work, you have to have a lot of twitches that add up together. There's, well, the way that twitches add up is called wave summation, but it could also be called temporal summation. So you have two types of wave summation. First you have unfused tetanus, also called incomplete tetanus, and then you have fused tetanus, also called complete tetanus. In your unfused tetanus, your action potentials are arriving. Every arrow on this graph represents an action potential arriving at the motor neuron. So activity at the neuromuscular junction, and then a new action potential spreading across the sarcolemma. So if they're arriving this frequently, about 50 action potentials per second, then you're going to have a twitch. You're going to have the relaxation period of that twitch, and it will not relax totally before the next action potential arrives and you have the next twitch. And then that twitch will start relaxing, but before it's able to relax all of the way, while there's still some calcium in the cytosol, you're going to have another action potential, more calcium released, another twitch beginning. So in unfused or incomplete tetanus, the twitches overlap. They don't overlap completely, and over time they add up to some maximum value. In fused tetanus, the action potentials are coming more quickly. They're coming at 80 to 100 action potentials per second. And so you get your first action potential that causes the first twitch, and then that twitch doesn't have a chance to relax at all. So you never have calcium levels drop in the cytosol, and then the next action potential arrives and you start another twitch. So even more calcium is released, you get even more muscle force, even more myofibril contraction, and then the next action potential arrives and then you have the next twitch begin until you get to the maximum force that that muscle cell can produce. And that's your maximum tension resulting from fused tetanus. So in order to get the maximum force out of your muscles, you have to have enough action potentials that you can get this fused tetanus. If you have incomplete tetanus or unfused tetanus, you can get a good amount of force, but not as much force. It will never be as much as what you can get from fused tetanus. In order to have your muscles contract in a way that you can actually notice, you have to have one of these things occur, either unfused or fused tetanus. Otherwise, it will never happen. And then finally, please note that there are other types of tetanus, and this is not those. So fused, unfused tetanus, that's referring to twitches in a muscle overlapping due to action potentials coming very quickly at the muscle. You also have the diseased tetanus, which is caused by a toxin from a bacteria. And then you also have spastic paralysis, which is also called tetanus. That would be when your muscles are contracted and they're paralyzed in that position so that you can't relax them. That's yet another type of tetanus. There's a relationship between the length of your muscles and the amount of tension that they can produce, or the amount of force that they can produce. You want to have kind of optimal overlap between the thick and thin filaments when you start the contraction to get the maximum amount of force. So if your muscle is lengthened, like this muscle will be lengthened if you have stretched it out by extending your palm like that. If the muscle is lengthened, then the myofibrils will be stretched, and your thick and thin filaments will only have a tiny little area where they're overlapping. If you start contracting at that point, there's not very many cross bridges that can form because there's not very many places where thin and thick filaments overlap. So you're not going to be able to make as much force. On the other hand, if the muscle is too short, if you're kind of putting your body in a position that squeezes the muscle to be shorter, then you're going to have a whole lot of overlap between the thick and thin filaments, and there's really nowhere for it to go from there. You can form a lot of cross bridges here, but if you're trying to move the thin filaments in towards the center, they're already at the center, so they really can't move very far at all, if at all, depending on how much the muscle has been shortened. So having the muscle at a neutral, relaxed position at its normal length, that is the way to get the most force out of the muscle contraction. That has kind of optimal overlap between thick and thin filaments, so that there's enough place where they overlap so you can get a lot of cross bridges, but there's also room to go from here, so the thin filaments have room to move in towards the center of the thick filaments. You also have a couple different types of skeletal muscle cells. You have type 1 fibers and type 2 fibers, and then the type 2 fibers also have subtypes. Your type 1 fibers can make kind of contractions that take a long time but last a long time. Your type 2 fibers are the stronger fibers. They can make really strong contractions, and it can do it very quickly, but it doesn't last for a long time. So your type 2 fibers are better for sprinting. It's a strong, fast contraction. Your type 1 fibers are better for a marathon, where it's not as much force, but you can endure and produce the force for a much longer time. One of the differences between those two types of cells is the ATPase on the myosin head. Each myosin head has an ATPase that is a part of the head that just lets it break ATP down into ADP and a free phosphate so that it can use the energy. So your myosin heads need to have an ATPase so that they can use ATP to let go of actin and to cock the head. So your type 1 fibers have ATPase on the myosin head that is it's a low activity ATPase, so it produces slower results. So the type 1 fibers will do crossbridge cycling more slowly because they're not able to turn over ATP very quickly. So they're not able to use it fast, so they have to go slow. Your type 2 fibers, on the other hand, have the high activity myosin ATPase, which allows them to use ATP very quickly. So the high activity ATPase will just very fast turn over ATP into ADP and phosphate and use the energy for something in crossbridge cycling. So that means that the type 2 fibers can do crossbridge cycling very quickly. Your type 1 fibers that have the low activity slow ATPase also are usually smaller in their diameter. Since they are smaller, they don't have as much room for myofibrils, so they don't have as many myofibrils in them. That means the total force they can generate is less. And they're also darker looking, so like dark meat would be meat that has a lot of type 1 fibers in it. Is darker because it has a lot of myoglobin, a lot of mitochondria, and a lot of capillaries. All of those things contribute to a dark color. So in this picture here, you have all these red cells would be the darker ones. Those are the type 1 fibers, and you can notice that most of them are a bit smaller than the pale cells next to them. That extra myoglobin, mitochondria, and capillaries means that they're making more ATP, they're getting more blood, they're storing more oxygen. They have more myoglobin to store more oxygen, they have more mitochondria to make more ATP, they have more capillaries to bring in more oxygen and more glucose. That lets them endure and contract for a very long period of time because they have a very high ability to make ATP. Your type 2 fibers, on the other hand, are bigger, so they're able to fit more myofibrils. That means they can make more force overall, but they don't have as much myoglobin, mitochondria, or capillaries, so that makes it lighter in color. So white meat would be mostly type 2 fibers. Since it doesn't have as much ability to make ATP or to store nutrients or bring in nutrients, you're going to have less endurance in the type 2 fibers. They can contract very fast and very strong with their fast ATPase and their many myofibrils, but they cannot contract for a long time because they don't have the ability to make enough ATP. Your type 2 fibers in general use more anaerobic catabolism than aerobic, or compared to the type 1 fibers at least. Since they don't have as much ability to get oxygen, they would be using the glycolysis and the creatine phosphate as energy sources instead. And you have two subtypes of the type 2 fibers, type 2A and type 2X, where the type 2X is just even stronger and faster than the regular type 2A fibers. In general, all of your muscles have all three types. So they have type 1 fibers, type 2A fibers, and type 2X fibers. But different muscles will have different combinations depending on what the muscle is usually used for. So muscles in your back, like the erector spinae muscles, those would be the iliocostalis, the longissimus. Those ones are being used all day long to keep you upright, and so those have a lot of type 1 fibers. On the other hand, the muscles in your arms and legs have more type 2 fibers. You're not using them like every second, you're only using them when you walk or when you pick something up. So they don't need to be as good at enduring long contractions, but they do need to be able to generate a lot of force when you need it. If you are exercising, you can shift what type of fibers your muscles have in them, depending on what type of training you do. So it's possible if you start out with kind of an even mix in your leg muscles between, well, it's mostly type 2 there, which it should be since it's your leg muscles, but also with some type 1. If you focus on a lot of sprinting, then you're going to end up with even more of those type 2 fibers. But if you focus on marathoning, you're going to end up with more of the type 1 fibers. So what you require of your muscles is determining what they can give you and what type of fibers they have. If you're doing exercise that has a lot of endurance, then you're going to get a lot more of the type 1 fibers that will let you be good at endurance. If you're doing exercise that's a lot more focused on speed or on strength, like weightlifting or sprinting, then you're going to end up with more of the type 2 fibers that will let you be strong and fast, but don't let you endure. In addition to wave summation, there's another way that you can increase the contractile force generated by your muscles, which is based on activating more motor units. So a motor unit is one motor neuron and all of the muscle cells that are contacted by its axons. So here in red you have the cell body of a lower motor neuron. Its axon exits in the anterior root of the spinal nerve and continues on ultimately to this muscle, and it branches and it contacts all of these three muscle cells. So these three cells and the motor neuron that controls them, those are all one motor neuron, one motor unit. If you were to have an action potential generated by this motor neuron, it's going to go down the axon, it'll go down every branch, and it'll get to each of these three cells basically simultaneousally, and they will all have a twitch at the same time. If you have multiple action potentials coming along this axon, then they're all going to go through wave summation, and they're going to end up in unfused or fused tetanus to give you, a contraction that has strength you can actually notice. Here's another motor unit right next door. All of these muscle cells are contacted by an axon, which comes from this blue motor neuron. And you can see that this motor unit is bigger. It has more muscle cells in it. So if it were to contract, it would generate more force. So you have a lot of different sizes of motor units. Some of them are very small, some of them are very large. If you're using the muscle for finely coordinated actions, like so your fingers for writing and typing and stuff, you're going to use a lot of small motor units for that, so that you can have a very fine-grained control over how much force your muscle is producing. If you're using the muscle for something a little bit coarser, then you're going to have really big motor units. So for instance, in your back muscles that are holding you upright all day, they don't have to do anything finely coordinated, they just need to keep you up straight. So they're going to have motor units that have like thousands of different muscle cells in each motor unit. Which means you have a lot less control over how much force the muscle can produce, but that's fine in this case. So one way of increasing your muscle force is the wave summation, but another way is by increasing how many motor units are active at any given time. So if you were to activate just the red motor unit, then you would have a little bit of force, but if you were to also activate the blue motor unit, you would have a lot more force. And that's called motor unit recruitment. So the more motor units you have activated in any given muscle, then the more force that muscle is going to produce. So you can be increasing muscle force by wave summation or by motor unit recruitment. Usually you're going to start by recruiting the slowest and the smallest motor units. The slow motor units would have the type 1 fibers. Since they're small, they would have few muscle cells in them. So at first, maybe you recruit this small motor unit, which can only generate a small amount of force because it only has a few muscle cells in it, and those cells are the small type 1 cells. Next, you're going to activate maybe a middle size motor unit that has bigger cells in it and a bit more of them. And then last, if you still need more force out of that muscle, you're going to activate the biggest motor units that you have in that muscle, which would be the type 2 ones that have really big fat muscle cells and a whole lot of muscle cells in every motor unit. So the more motor units you have activated and the more quickly each of those motor neurons is signaling its muscle cells with action potentials, the more force you're going to get out of the muscle. And you've got 3 basic types of contraction: isotonic concentric contractions, isotonic eccentric contractions, and isometric contractions. The isotonic concentric contraction is the basic type of muscle contraction that you're used to. That would be the type that you would use to lift a load. In the isotonic concentric contraction, the muscle is generating more force than the load, which means that the muscle is going to get shorter and the load would be lifted. It's concentric because the muscle is getting shorter. It's isotonic because you're generating a constant amount of force. When you're kind of ramping up the contraction, then you're going to be, generating more and more force until you reach the amount of force that you need, and you're just going to stay there at that amount of force and lift the object. An isotonic eccentric contraction is almost like the opposite. It's a weird contraction. Even though the muscle is contracting, it's going to get longer. It gets longer because the load is greater than the force generated by the muscle. So if you're holding this weight up here and you relax your bicep a little bit so that it's not generating as much force, then maybe it won't generate as much force as the load, and your arm would drop back down. It's called isotonic because however much force you're generating is the same throughout the contraction. It's called eccentric because the muscle is getting longer, not shorter. These are very interesting contractions. They don't use as much ATP. Since the muscle is not making as much force as the load, it doesn't cost as much ATP. But actually, the amount of force that it generates cannot be explained by the amount of ATP that it uses. It doesn't use enough ATP to explain how much force it's able to generate. And they're really not sure how force is being generated in the isotonic eccentric contractions, but it seems that it's relying on the elastic myofilaments in some way. So we used to think that they're just for structure, just holding the myofibrils together, but now it seems like they have a role to play in contraction in the isotonic eccentric contractions. What it is, we don't know exactly. The isotonic eccentric contractions are really good for people who need to do exercise but have to be careful not to strain their bodies. So that would be like, for instance, someone with a bad respiratory condition or a heart condition where they're limited in how much ATP they can make. They have to do something that will not require too much ATP. But for all those people, doing some exercise is going to be really helpful for their condition. So the isotonic eccentric contractions can let them do some exercise without stressing their systems too much. On the other hand, it is more likely for you to damage your muscle in the isotonic eccentric contraction because it involves this weird thing where your muscle is trying to shorten but it's actually stretching instead. So that just places a lot more stress on the myofibrils. So you have to be careful when you're doing them, not to overtax yourself. It actually seems that these are better for building strength. Like, you'll build strength more quickly if you're focusing on isotonic eccentric contractions. And it's also extra good for bone remodeling, for signaling your bones that they need to be stronger, because you're putting a huge amount of force on the bones. With the muscle, let's see if I get my pointer back. The muscle is trying to get shorter, so it's pulling on the tendons. But the load is also pulling. It's pulling the joint kind of straight, and that means that this tendon is getting stretched on both ends. And it's that stretch in the tendons that signals the bones that they need to be stronger. Then finally, you have isometric contractions. In the isometric contraction, the muscle does not change its length. So that means that the force the muscle is generating has to be equal to the load that it's holding or supporting. So that would be the type of contraction that you would use to hold your arm out and be holding something. The force that you're generating does not stay the same. It's going to vary depending on how much the load is. So it will always just equal the load, but if the load changes, then the force will change to match it. The muscle length is the thing that doesn't change in the isometric contraction. And exercise. Since we're talking about muscles, it's a good time to talk about exercise. So you've got two basic types of exercise: endurance training, which would be aerobic exercise, or resistance training, which is weightlifting. If you're doing endurance training, then you're favoring the type 1 fibers, so your muscles will end up with more type 1 fibers. Those are the slow ones that have a lot of ability to make ATP. Doing the endurance training is going to be good for increasing the blood supply to your muscles, and yeah, just increasing their ability to make and use ATP. Well, to make ATP and to bring in oxygen and nutrients. It has giant health benefits for essentially every system in your entire body. It has really important benefits for the cardiovascular system, but it also important benefits for the immune system, for the brain, for the nervous system, really for every system. It's good for the whole body, you guys. We really don't understand why it's so good for you, but it really, really, really is. It's amazingly good for you. And especially if you maybe don't have health insurance, this is what you should probably be doing to keep the doctor away. It's between the endurance training and having a good diet and sleeping enough, those are the three things that you need to do to take care of yourself and maintain your health. Then you also have resistance training, which favors the type 2 fibers that are fast, that give really strong contractions, but that can't contract for as long. So if you're doing the resistance training, you're not going to get better at bringing in oxygen to your muscles, but you are going to make them bigger and stronger. So you actually would never actually add more muscle cells. You have however many you have, you don't get more. But if you start out with, you know, your regular size muscle cell and then you start lifting weights, you're going to add more myofibrils in there. The muscle cell will start manufacturing more myofibrils. It'll actually, the whole cell will become bigger and fatter and stronger. So it's going to make you a lot stronger. That's good for your joints. It helps stabilize your joints. It's also good for your bones. It helps signal the bones that, you know, they do need to be strong. They do need the calcium. And so the weightlifting is an important part of exercise. But just make sure that you're also doing some aerobics as well, because it's the aerobics that has benefits for every body system, not just a couple. When you first start exercising, you're going to notice that you're getting stronger pretty fast. That's not actually because of changes in your muscles, it's because of changes in the motor neurons that are supplying those muscles. The motor neurons will actually get faster and better at signaling the muscles. And you'll get more coordinated so that you can contract different motor units in a more synchronized fashion to give, more powerful contractions. Then after that, you'll start adding, myofibrils to the muscles and make actual changes in the muscles themselves. And yeah, it's good if you're doing exercise, whether aerobic or weightlifting, it's good to also get some stretching in there so you keep your joints limber. And with any exercise habit, it's important that you be consistent and that you go slow so you don't hurt yourself. It is possible to damage yourself by exercising more than what your body is able to withstand at that time. But as you exercise a little bit, then your body's endurance will build. It will be able to take more strain, and then you can exercise a bit more. So just, kind of pay close attention to your body. If there's kind of an unusual pain, even if it's a subtle pain, even if it doesn't hurt a lot, even if it's only a little bit, but it's, it's weird, then you should probably stop that and it could go a little bit slower. What really matters is that you're consistent. So if you're going to do it 3 times a week, then do it 3 times a week. Even if it's like a bad day and you're just extra tired that day, just do something really light, but maintain the habit. One more thing I was going to say. Oh boy. Oh yeah, so you can always kind of know how much strain you're under, at least aerobically or cardiovascularly, by paying attention to your heart rate and your breathing rate and your sweating. Right, so I guess you'll probably be able to feel your heart kind of pounding, but if you're sweating and your breathing is becoming labored, so you're breathing faster and deeper, that means that you are activating your cardiovascular system to supply more oxygen and glucose to your muscles. So that means that you are doing good aerobics there. If you're trying to do aerobics but you're not sweating and you're not breathing hard, then you need to increase what you're doing, because you're not putting a stress on your cardiovascular system. You're not letting it know that, that more is needed, more ATP is needed. Okay, so that concludes everything we're going to say about the skeletal muscles. Now we're going to talk about smooth muscle and cardiac muscle. First, cardiac muscle. So cardiac muscle cells have a special name. They're cardiomyocytes. They're kind of short and squat, like this guy is. They are branching, and they connect to each other at intercalated discs that have gap junctions in them. So here you have an intercalated disc. These are gap junctions. You also have desmosomes there. So the desmosomes would be giving strength so that these cells are strongly stuck together. The gap junctions are letting ions flow through. So calcium and sodium can move freely from one cell to the next. That means that if you have an action potential in one of these heart muscle cells, it's going to spread to all of them through the gap junctions. Inside of the cells, you also have the myofibrils. So these kind of river-like things. These are the myofibrils. Here's a myofibril with the myofilaments shown. So the myofibrils will actually kind of cross these intercalated discs. or at least if you have a myofibril in this cardiomyocyte here, the next cardiomyocyte will have another one in the same location. And you still have the sarcoplasmic reticulum around those myofibrils and the T-tubules. You have a little bit less than what you have in the skeletal muscles, but you still have it there. You have a lot of myoglobin in your cardiomyocytes and a lot of mitochondria, so they have a very high ability to make ATP and to store oxygen, which is really important because the heart can never take a break. It doesn't ever get to rest. They also have specialized cardiomyocytes that generate the action potentials that cause the heart to contract and also distribute them across the heart. That would be the heart's conduction system. So all of this, I mean, I guess the heart would be there too. It's not really shown, but all of these yellow guys are special cardiomyocytes that are conducting action potentials to the entire heart and also generating them. So here, especially in the SA node, the sinoatrial node, you have specialized cardiomyocytes that generate action potentials rhythmically, kind of spontaneously on their own. And then those action potentials would be carried to the whole heart by the rest of the conduction system. So if you don't have any signals from the brain at all, then you'll still have action potentials in the heart coming from the sinoatrial node. The natural rate at which the sinoatrial node generates action potentials varies depending on the person, but it's about 100 per minute, which would give you about 100 beats, 100 BPM, 100 heartbeats per minute. That's pretty fast, actually. Your resting heart rate should definitely be lower than that. That's because the parasympathetic nervous system is always signaling the SA node to slow down. It's always slowing down your heart rate at rest. Then if you inhibit the parasympathetic nervous system because you got scared or you're working out or something, then your heart rate will increase instead. For your smooth muscle cells, they have a completely different structure from cardiomyocytes or the regular skeletal myocytes. They still have thick and thin filaments, but they are not arranged into myofibrils, so they do not have myofibrils. The thick and thin filaments would actually be running across the cell in like this diagonal crosshatch pattern. Every kind of red line here would be a thick filament surrounded by thin filaments on all sides. So if you zoomed in on it, here's your thick filament in the middle and your thin filaments on either side. Actually, they would be kind of just around it in a circle. Then you have these points on the cell membrane called dense bodies, where those myofilaments are anchored. So that if the muscle cell contracts, you're going to have the thick and thin filaments sliding past each other. Since they're all arranged in this diagonal pattern, instead of the cell just getting shorter, it's going to bunch up like this. If you look at the thick filaments and the thin filaments, their arrangement is different from how it is in the skeletal muscle cells. So your myosin heads are distributed all along the whole length of the thick filament, not just on the ends. And they're on all sides. They have their hinges go in the opposite direction on different sides, so their orientation is opposite on different sides. Then the actin, or the thin filaments, is just a string of actin. You don't have the troponin or the tropomyosin blocking the active sites. So it's always possible for the myosin to make a crossbridge with the actin. If the muscle contracts, then your myosin will grab the actin, it'll do the power stroke. That'll be happening in opposite directions on each side of the thick filament, and the thin filaments will slide past the thick filament on either side. So the whole thing will get shorter. Apart from that, smooth muscle cells don't all share the same characteristics. The way they contract is basically the same for all of them, but you also have a lot of characteristics that are different for them, depending on where the smooth muscle is and what job it's going to do. So for instance, the little erector pili muscles, which are the ones that make your hair stand up on your skin. Those, of course, are different from the muscles in your sphincters that need to be contracted all the time. Some of these smooth muscle cells have gap junctions and some of them do not. Some of them respond to lots of different stimuli. Some of them only respond to neurotransmitters. In general, smooth muscle cells that have the gap junctions are more likely to respond to multiple stimuli than the other ones. The contraction for smooth muscle is quite different from skeletal muscle, as you might expect from the different structure of the cell. For excitation, in skeletal muscle, you have acetylcholine released at the neuromuscular junction. In smooth muscle, it doesn't have to be a neurotransmitter that stimulates the cell. It could be acetylcholine, or it could be epinephrine, or norepinephrine, or it could be something else entirely. It could be a hormone, or even something else like stretch. So here's kind of an example of some smooth muscle. These cells are connected to each other by gap junctions, and then you have an axon passing by, and the axon has little bulges on it called varicosities. So you notice this axon is not actually contacting the smooth muscle at something like a synapse. So there is no like neuromuscular junction between smooth muscle and a motor neuron. In this case, this would actually be a postganglionic neuron. So instead, you're doing a different type of transmission entirely, and you don't have an axon terminal where these neurotransmitters are being released. They're released from all these varicosities, these little bulges along the axon. This neurotransmitter would be acetylcholine if this is a parasympathetic postganglionic neuron, or it would be norepinephrine, probably if it was a sympathetic postganglionic neuron. These are called synapses en passant, or synapses in passing. The little bulges are called varicosities. So you have neurotransmitter released from these varicosities, and it's going to diffuse onto the smooth muscle and bind to receptors there. It's not going to be contacting one specific cell because of the distance, because it's not actually contacting one specific cell. The neurotransmitter is just going to diffuse to all of the cells in the area. And it's going to cause local potentials after binding to the receptor. You can have lots of different types of receptor on the smooth muscle cells. You're going to always have a local potential in response. That could be hyperpolarizing, or it could be depolarizing. And if you get enough of them adding up, then you will start an action potential. And then that action potential, in this case, could travel to all of these cells through the gap junctions. So the neurotransmitters would be either acetylcholine or norepinephrine. The receptors, if we're talking about receptors for the neurotransmitters, then they would be either muscarinic acetylcholine receptors, or they would be alpha or beta adrenergic receptors for norepinephrine. All right, the parasympathetic nervous system uses acetylcholine and muscarinic acetylcholine receptors. The sympathetic would be using norepinephrine and alpha or beta adrenergic receptors. So that's excitation. Then in excitation-contraction coupling, you would have that action potential spread across the entire sarcolemma, and if you have gap junctions, it'll also spread through the gap junctions to the cells that it's connected to. But you don't have T-tubules in your smooth muscle. You do have sarcoplasmic reticulum, just a little bit of it along the edges of the cell, and no T-tubules at all. But you do have the voltage-gated calcium channels in the sarcoplasmic reticulum that are linked to the cell membrane or the sarcolemma, and you also have more voltage-gated calcium channels in the sarcolemma itself. So when you have action potential spreading across a cell, that is going to activate those channels, and calcium will enter the cell, either from outside the cell or from the sarcoplasmic reticulum into the cytosol. So the end result of excitation-contraction coupling is the same, where you have calcium entering the cytosol. Then contraction. The contraction phase is fairly different because you don't have troponin or tropomyosin. So instead of calcium binding troponin, once calcium enters the cell, it would be binding to calmodulin. And then that complex of calcium with calmodulin is going to activate the myosin light chain kinase, or kinase, also called MLCK. MLCK is going to activate the myosin head ATPase, and then you get your crossbridge cycling. So here's the diagram of that. Here you have your voltage-gated calcium channels that have been opened by the action potential. Calcium enters the cell from outside, or it enters the cytosol from the sarcoplasmic reticulum. Then you're going to have it binding to calmodulin right here. So this complex of calcium with calmodulin activates MLCK, or myosin light chain kinase, and then you have the active myosin light chain kinase, and then that active MLCK is going to activate the myosin head ATPase. The ATPase on these myosin heads that allows them to use ATP. Once they're able to use ATP, then they're able to cock, they'll bind to actin, they'll do the power stroke, they will use ATP again to let go of actin, and repeat. So the crossbridge cycling is the same, but the way that you initiate crossbridge cycling is different between smooth muscle and skeletal or cardiac muscle. Then the relaxation phase is, of course, different as well. The relaxation phase would begin when your calcium levels in the cytosol drop because it's being pumped either back into the sarcoplasmic reticulum or back out of the cell. As the calcium levels drop, calcium is going to let go of calmodulin. Once calmodulin is no longer attached to calcium, then that's going to inactivate the MLCK. Once the MLCK is inactivated, then the myosin head ATPase is going to be deactivated as well. You also have another enzyme called the myosin phosphatase that will also be deactivating the myosin head ATPase so that it deactivates more quickly. Once you have the myosin head ATPase deactivated, it can't use ATP anymore, and so you will not have crossbridge cycling occurring anymore, and the muscle will relax and go back to its original size and shape. Some smooth muscle cells, specifically the ones that the ones that form sphincters, have a special state they can enter called the latch state. In the latch state, basically everything gets turned off while the cross bridges were still formed. So normally you would have myosin use ATP to detach from actin, and then you would deactivate the myosin head ATPase to relax the smooth muscle. In the latch state, you would have the myosin head attached to ATP, and then you deactivate the myosin head ATPase. Since it needs to use ATP to detach from actin, and it can't use ATP anymore, it's going to have to remain attached to actin in the same position so that the muscle cell will not actually relax. The muscle contraction has relaxed, but the muscle cell has not returned to its original length. It's going to stay shortened. That lets your sphincter muscles stay closed without using ATP constantly. So you can just kind of set it and forget it. So you've got many types of smooth muscle, but a couple types would be the single unit smooth muscle and the multi-unit smooth muscle. In single unit smooth muscle cells, or smooth muscles, all of the cells are connected to each other using gap junctions. So if one of them contracts, the action potential will spread to the others and all of them are going to contract. You find that in a lot of different places, but prominently all around the intestine, in the wall of the intestine and the wall of the stomach and the esophagus, the entire gastrointestinal tract. The smooth muscles in the wall are connected by these gap junctions that lets them contract as a single unit. On the other hand, the multi-unit smooth muscle cells are not connected to each other by gap junctions. Each one has to be stimulated independently of the others. You find those wherever you need kind of finer control over the muscle contraction, but the muscles don't need to work together quite as much. So, for instance, in the eye, the muscles that are controlling the shape of the lens, which controls how light is focusing on the back of the eye, those are multi-unit smooth muscle cells where each cell needs to be individually stimulated.